Okay, hello. My name is Phillip de Lorimier. The date today is the 17th of April, 2020, and I'm going to be talking a little bit about my senior thesis project, which is entitled Characterizing Soil Microbial Community Response to Prescribed Fire Along a High-Resolution Soil Depth Profile. At the time of this recording, my project has not yet finished due to the situation with campus being closed. So hopefully I'll be able to give this project some context, explain where I am in the project, and some of the future directions that I hope to take. So by way of introduction, I suppose I should introduce myself a little bit and my interests. Here at Berkeley, I'm a senior graduating this semester, but here at Berkeley, I've been able to kind of explore three areas of ecology that have really fascinated me, and luckily I was fortunate enough to be able to combine them into the senior thesis. So those three are soil science, fire ecology, and microbial ecology. And I think each one of them, the intersection of those three, there's a really kind of cool cutting edge of science going on. And here at Berkeley, those three disciplines really do combine in a very cool way. I worked in the lab of Dr. Matt Traxler under the supervision of Monica Fisher and Neem Patel. And I was part of the Pyro team. So hopefully I'll be able to explain a little bit about that larger project for which my project was kind of a sub-project of. To start off with a little bit of introduction on soils, microbes, and fire, I think it might be useful to start with kind of a broad context of global change and land management at a broad scale. So as we all know, due to climate change and increasing temperatures, it's part of the story why we're seeing this huge increase in wildfire frequency. This figure on the left is from a paper that was, very prominent back almost, 2 decades ago now, I guess, which pointed to the role of climate change in increasing wildfire frequency in the Western United States. That is to say, climate is a part of the story. It's not the only part of the story. Here at Berkeley, there's been a lot of research by Scott Stevens' lab and others to look at the role of past land management on the trajectory of forests in the United States, specifically the policies around fire suppression, which are a big part of why we are seeing such catastrophic mega fires in California and Australia, and those can have really profound effects on ecosystems. But kind of going down to the finer scale, there are a lot of cool basic science questions that have yet to be explored within the role of microbes in these forest soil systems. We know that microbes are, play a huge role in these global biogeochemical cycles, in water cycles, in basically in plant and forest health. And so it's important to consider them and how they might be impacted by these, huge disturbance events. So there are a lot of basic science questions to be uncovered about disturbance and the role of microbial succession. And soil, of course, just kind of provides this really unique matrix for it all. I'm going to zoom in here on the bottom left. This graph shows kind of an idealized curve of soil temperature throughout the course of a day. And you can see that, as temperature fluctuations are greatest at the surface and they decline with depth. And so when we're thinking about fire as a disturbance, inputting temperature into the soil, we can think of it as a very spatial directionalized disturbance. And so we think that the effects from fire are going to be greatest at the surface and decline with depth. And that might seem like an intuitive hypothesis. And that's kind of coming from both the physical side of the heat, but also the chemical side. Fire is known to restructure chemical compounds in the soil, creating pyrolyzed organic material. And those new compounds, can only be accessed by a certain group of microbes. So maybe the effect of the chemistry, post-fire chemistry, is going to be visible. So that's kind of the general outlines of my project. I was curious to understand how soil microbial communities, which I define here as soil bacteria and fungi, are affected by a low severity prescribed fire, which is an emerging tool in wildland resource management to be able to counteract some of those trends which I spoke at the beginning of this video. Again, the Stevens Lab, Rob York, many people have been involved with prescribed fire research at Berkeley, and I think it's an exciting time to be in this field and be looking at the effects of another side of prescribed fire on the soils and microbes. All right, moving on now to the methods of my project. So I'd like you to focus, I guess, right now on this diagram here, which is kind of a schematic of my sampling design. Samples were collected at the UC Blodgett Experimental Forest, which is located just up the mountains in kind of the El Dorado National Forest, or within nearby the El Dorado National Forest. The nearest town is Georgetown. It's kind of a mixed conifer forest, and the soils are very rich in organic matter, and that'll play a role in a little bit. So the sampling design that I eventually settled on with Monica and Neem was looking at this very highly resolved spatial profile within the soil profile. So you can see here that I collected samples from 0 down to 20 centimeters. The first 5 samples are at an interval of 1 centimeter. Then there's a 5 to 10 centimeter chunk, and then 10 to 20. So that was kind of the rough, overview of samples. And so there's 7 samples for each time point. And then you see here this this rain cloud is depicting a huge impulse of precipitation, which happened in late November of 2019. We were hoping to do the prescribed fire in November, but due to some of the larger conditions that were going on at the state, it was not considered safe to do that. And so instead we sampled in December, and so we had this kind of pulse of precipitation right before we sampled. So we kind of thought of it as a treatment of this, of a kind of disturbance of precipitation on the soil. And then luckily in February of 2020, we were able to do the prescribed burn and collect another round of samples. So you see we have kind of this this succession of 2 discrete disturbances, one being a pulse of rain, and then kind of the cumulative effect of fire. So it kind of, the way, just by chance, the fact that we weren't able to burn the fall actually kind of ended up adding a nice dimension, I think, to this study. So that's the outline. You can see here over on the right side, this is a picture of the soil core, this like long cylinder of soil, and I divided it up into those segments and put them in test tubes, took them back to the lab. So that's kind of the field work that was involved in this study. And there's some pictures of the fire below. We'll get to that in a second. Figure 4 shows kind of the overview of the bioinformatic pipeline that I employed. So the soil samples back in the lab, first of all, we got the pH on them, and then extracted the microbial DNA using the QIAGEN DNeasy soil test power soil kit, which basically takes all the DNA from the fungi, microbes, and bacteria out of the soil, which we were able to amplify. And this is kind of this is the step where we were doing right before the campus was shut down. So this is kind of the point where we were on pause. But what will happen when we return is amplification of the microbial DNA, and especially these kind of key genetic markers, the 16S gene for bacteria and the ITS gene, which will allow us to identify fungi. So this kind of schematic shows like the pipeline of extracting DNA, and then and the point here where we separate between bacteria and fungi is the choice of this genetic marker, either the 16S gene or the ITS gene. this whole process was is fairly time consuming and an expensive process, no doubt. But it was, I was really happy that I was able to do such a detailed look at the first, first few profiles of the soil column, and hopefully that there will be some good data on that coming up. So that's kind of the first part of the experiment. And then I had kind of a sub question going into this. You mentioned my hypothesis was that the effect of fire would be greatest in the top few centimeters. But also what we have in the top few centimeters is an organic soil horizon, known as the O horizon. When you look at soil profiles, it's composed of litter and decomposing organic matter. So you can see in this picture here, these pine needles and kind of the first bits of, decomposing organic matter. That is the O horizon. We did not sample from this horizon for a couple of reasons. First, it was suspected that this layer would be burned off completely in the fire. We'll see later on that that was not the case. But also this community is composed largely of decomposers and bacteria that live on leaves that, maybe are not as reflective of the overall soil community. So that was kind of the rationale, I suppose, for why we did not sample from the O horizon. But I thought it was important nonetheless to at least get a metric of how thick the O horizon was at each of the time points to see if it had any kind of insulating effect on the microbial communities further down. So that was kind of a little sub part of this project. And then finally, the moment we've all been waiting for, in February 2020, like I said, we got to do a prescribed burn, which was really exciting. If you've never been in nearby, like a kind of, it wasn't like a massive wildfire or anything, but it was still, the flames were, I'd say, between, 2 to maybe 5 feet at the max. It got pretty hot, and it was really exciting to see, after a long period of waiting, to see it come into fruition. So here we have on the left picture of these two, you have the prescribed burn going through the plot. And then over here on the right, this is a few days after, you can see the layer of ash and char coating the surface. So this is kind of what I was talking about earlier with the pyrolyzed organic carbon. That is, what is in that that white layer here, and presumably affecting the microbes below chemically. All right. That is the sample, this kind of methodology I took to this project. And now I guess let's look at the results that we got. All right, on the left side here, one last thing to mention is that before we did the burn, we inserted these thermocouple probes to measure soil temperature at the same depths at which we sampled. So that's 1, 2, 3, 4, 5, 10, and 20 centimeters. With the intent being to correlate the temperature at which the soil was exposed to the subsequent dynamics, the microbial dynamics. So you can see here on the left that one thing that was really noticeable was there was a remarkable heterogeneity of fire heating effect on the plot. So these two plots show two different data temperature probes, but that are really close to each other, probably less than 5 meters away from each other. And in the top one, you see that there was this huge spike of temperature right at time 0, which is when the fire occurred, and then leveled off after that. But then in the bottom part here, data logger F, you don't see that same spike, and in fact, it looks as if there's no real significant effect of that fire. That's a really interesting finding, and one that was repeated across the other thermocouple probes. There are more that I did not include in this poster. So that's kind of the first clue that there is going to be some kind of maybe some interesting results. Moving on now to the next graph, the O horizon thickness. the kind of thing that jumped out to me immediately was that over here on the right side of the graph, I guess I should say this graph shows the thickness of the O horizon that I measured above. There's a control plot, which is in green, and a burn plot, which is in this kind of orangey red. The fire, which happened here in February, did not really remove all of the O horizon like we had initially suspected. So that is, kind of a noteworthy finding, and one that might, play a role in the effect on the community composition of the microbes. So there's a lot of spread in this data, of course, and so it's tough to take away any kind of conclusions, but that is one thing that is definitely noticeable. And then over here is the pH data that we collected. The only other kind of hard data that we have at this time, but it's there's also a lot of spread in this data. I think the one thing to take away is the effect of the rain here in between October and December had an effect across the board of moving the pH to the left. So you can see in October, it's, closer to 5.5, that gets shifted down to 5 here in December. The way to read this graph, it's a little tricky, I guess, is the pH is displayed across the top, which is a measure of soil acidity, more acid being the lower end, basic being on the upper end over here. And then down the y-axis is depth. So you can think of it as you're looking at a soil profile, the surface is here at 0, and then it goes down to our 20 centimeter measurement. Again, as before, green is control, it's a little tough to see, and red is burn. So there's the control plot and the burn plot. The effect of the fire on pH was one that we expected to be more pronounced than it was. We would have expected it to be a shift of increasing pH, because part of those pyrolyzed organic carbon is a lot of basic compounds, ones that move pH upward. However, there really was not a significant effect doing an analysis of variance on that data. So that's kind of an interesting, thing to note as well. Taken all together, what we can say in terms of discussion on the results and some initial speculations, I guess, that we have, is a strong effect of seasonality of the disturbance on the soil. And what I mean by that is the seasonality of fire played definitely a big role. When I was there and speaking with, Ariel Thompson and Rob York, who are two of the coordinators of the prescribed burn, they were mentioning how the fuel that is consumed during the fire was significantly more damp in that February burn than it might have been in, say, if the burn had happened in October. And that is something that's definitely probably that most likely had an effect on the temperature. Why it was so heterogeneous. Some places where the fuel was not completely dry, and as well as some of the chemical effects. Maybe there wasn't a complete combustion of the fuel. So seasonality of the burn definitely had a big impact. Seasonality of the disturbance in terms of precipitation, most definitely. After a long dry season, we didn't get too much precipitation in the few months before November. There is kind of there's a big impact that has at that certain time of the year. One thing that I'm beginning to suspect is that the O horizon, at least in the case of this prescribed burning, may actually be a buffer to some of the changes of community composition that we would expect from a larger fire that consumes the O horizon. We saw how there was a good amount of organic material left behind after the fire, and it's most likely that it provided kind of a buffering effect. That being said, really the point of this project was to look at the microbial communities, which we were unfortunately have not been able to do as of yet. We are working to do so when we return to lab And there's going to be a lot of really cool data analysis ahead of us. And actually, at this point, I will go to the next slide to show some of the possible kind of, I'm not going to walk too much through this slide, but suffice to say, this is from a collaborator at the University of Wisconsin with some folks in the Traxler lab. They're collaborating on this large project with fire and soil microbial community dynamics. This is one of their papers looking at, any number of things, the effect of pH and organic carbon on the distribution of communities. Over here, we have kind of a breakdown on a phylogenetic level. And then over here, a cool kind of co-occurrence map, co-occurrence network of soil microbial communities. So there's a lot of really interesting software out there that's enabling us to look at relationships between the microbes. And I'm excited to see what we have, and if it turns out that there isn't a huge impact, maybe that's going to be a good thing to say for the safety and environmental responsibility, I guess, of prescribed burning, that it is an appropriate management tool with respect to soil microbial communities. So that's kind of what's to come. It's been a great experience doing this research project. Monica and Neem, I am very grateful to them for their mentorship and just all around awesomeness, helping me through it. Of course, Dr. Traxler for his support. And I'd also like to thank the sponsored projects for undergraduate research and the Regents and Chancellor's Research Fellowship for grant funding to help me pursue this project. It's really been, something I've hoped and dreamed about doing for a long time, and it's a great feeling to be able to have something of my own to be able to present. So hopefully you took something out of this presentation, and I hope you enjoy their other presentations.